Hello, everyone, and good afternoon. Thank you for joining us for the virtual 9th Annual Forensic Science Symposium Lunch and Learn poster session hosted by the Global Forensic and Justice Center. Here's Joseph with his presentation. It's all yours, Joseph.
Hi, everyone. My name is Joseph, and I'm a graduate research assistant working with Dr. Deetta Mills at FIU, and I'm working on alternative collection methods for touch DNA. So I'll give a little background and then get right into my approach in this project. So the sensitivities of DNA technologies have continued to increase, and forensic laboratories are being expected to yield more probative DNA profiles with less and less collected biological material from crime scenes. The most commonly encountered biological material is known as touch DNA. And this is composed of epithelial cells left behind in the sweats and oils found on the hands of individuals, which is then transferred to surfaces and objects at crime scenes. So touch DNA evidence should be treated like trace evidence because there's no way that you can know how many cells are deposited into or onto a surface. So in a traditional DNA workflow, we encounter issues like the inadequate collection by the standard method, the cotton swab, and the loss of DNA during extraction. So with those two main problems, when we think about approaching a solution in the context of collecting touch DNA samples, we have to think about the factors that affect the collection of touch DNA across various surfaces, such as the roughness and the porosity of the surface. So there's been intense focus on the downstream processes of DNA, like getting better extraction efficiencies and having so many different kinds of amplification kits. However, the DNA loss from extraction and stochastic effects during amplification, they all persist in touch DNA samples as they move down the workflow. Now, there are gaps in this front end called collection. So I'm focusing more on that in this poster. And with cotton swab collection, it's fast and it's easy, right? There are practical reasons why we like it. But it does present two major issues. Cells become trapped in the cotton matrix and are not released effectively during extraction, and the cotton tends to rip and tear on rough and porous surfaces, especially when the swabs are pre-moistened. So it's also worth mentioning that swabbing in general for touch DNA is destructive toward any forms of evidence that also may be present, like fingerprints. So it's for this reason that vacuum collection was selected, so that this can be a non-destructive method as it continues to be optimized. So let's dive right into what I did here and the materials and methods. What I'm comparing to the cotton swab specifically here is the Venturi vacuum device. And what that is specifically is this fancy pump, which you can insert compressed gas. And in this scenario, I'm using CO2 so that my device can be portable. But the compressed gas will go through the pump and generate a suction, which I then can use to collect DNA. I connect it to a series of tubes and then to a syringe apparatus, which is completely disposable. And that is outfitted with an alternative collection matrix, polycarbonate filter. And the difference that polycarbonate filter has between cotton is that when you collect with it, it adheres to cells as opposed to absorbing the cells and trapping them within the matrix like cotton does. So to test these methods, I created a cell suspension from buccal swabs. I took the cells off of the swabs and put them into a solution and calculated that solution so that I could deposit approximately 100 cells onto surfaces and then use my two collection methods to collect with them. So after I did that, I allowed the cells to dry onto the surfaces, and I collected them with wet and dry treatments for each collection method. So at this point in the DNA workflow, I didn't want to take my samples through extraction because it introduces all this variation that I wanted to eliminate so that I could adequately compare my two collection methods. So I opted to direct amplify my samples with 6 core loci so that I could form my mock DNA profile. And I analyzed the samples with capillary electrophoresis. So if we look over here at the results on the right side of the screen, we can see relative fluorescent units on the y-axis, and the x-axis has our different treatments of cotton dry collection, cotton wet collection, PCF dry collection, and PCF wet collection, as denoted by the different colored bars. We have the green as the cotton dry, the yellow is the cotton wet, and the 2 blue are the dry and wet for the polycarbonate vacuum collection. And so each grouping of bars represents loci, and so I can compare each loci with per each collection treatment. So as you can see, most of our loci give a relative fluorescence units over our threshold of 150 RFU, which is denoted by the red line. And some things to note here is that for the cotton collection, the dry collection yielded much better peak heights than the cotton wet collection, which was surprising to me due to most touch DNA scenarios. We use pre-moistened cotton swabs to do our collections. And then, as you can see, the polycarbonate filter combined with the vacuum and either wet or dry yielded the highest peak heights, although there was a bit more variation with those bars, although the variation has more to do with the amplification. And finally, there was some dropout of the D16 loci, so I completely lost that in my cotton wet collection, which was also very surprising. So as I move forward with these results, I will be testing off of various brick materials, traditional red brick, cinder block, and other types of porous blocks in between. Typically those found in people's driveways. And with these results, I hope to also optimize collection protocols for porous materials that are also more challenging, like rope. With that, I will take any questions if there are any questions yet.
Thank you, Joseph. We actually do have one that just came in. If anybody else has questions, just put them in the chat. You can send it to either myself or directly to Joseph, and we will get to those. This question says, impressive work. Are you intending to test different filters?
So when I started all this work, I actually had 3 different alternative matrices or filters, but they were just too fragile to put on the front of my vacuum device. If I were to use my other possible filters or membranes or anything like that, I would want to use it in a further prototype that protects the matrix so that it's not just exposed on the front end, because it was very hard to collect with like membrane type filters, which I had been testing on rough surfaces. So Yes, that is a consideration, but not on this current prototype of the Venturi vacuum device.
Joseph, could you explain the graph real quick? I can't read the bottom of that. Could you maybe just explain what each
Sure.
x-axis is?
So, what was the last part you said?
Sorry, could you just explain what each thing is on the x-axis?
So on the x-axis, we have here these different groupings of bars, and each grouping is a core STR loci. So we have chromosome 5, chromosome 13, chromosome 7, chromosome 6, chromosome CSF, and penta D loci. So for each of those loci, there are 4 collection treatments, and that is what you see on the x-axis.
We have another question here that says, have you had any results on rough surfaces? You may have touched on that earlier.
Yes, I do have results from rough surfaces, although I know that they can be better, so I chose not to show them with this these results. If I had shown them, it would have only drove home the point that as we move from smooth to rough surfaces, collecting DNA and recovering that DNA from whatever matrix we're using to collect becomes all the more difficult. So in terms of improving the results so that I can actually compare them, I would have to increase the amount of aqueous solution that I do in a wet collection, and that does affect the tubing and the contamination of the Venturi pump if I decide to use more solution when I'm collecting, which is necessary when you're collecting from a porous surface like brick. So until I figure out how to do that without contaminating the vacuum pump, I will not show the results.
Okay, thank you. We have another question here that says, what brand swabs were used in the cotton swab collections?
I would actually have to ask my lab manager about that and get back to you on that.
Okay, so whoever asked that, I would suggest just checking up on the written answers, and we can provide that later on.
Right, that is a good question. Important to know.
Joseph, one question. Was there anything that you were surprised about in this study?
Yes, I was surprised at how poorly the wet cotton collection method did on the steel bar, which was not even a rough surface. So I wasn't expecting it to do so poorly because we use pre-moistened swabs as the standard collection method for touch DNA. So what I did here when I pre-moistened the swab, I pipetted 20 microliters of DNA-free water onto the swab, and then I did my collection with it. And that very much surprised me. So because I saw that, I am working on adapting the cotton when I compare the cotton to the vacuum, doing the double swab method, in which you pre-moisten one swab and you take sampling and then you go over it with a second dry swab and collect the leftover moisture. And I think with combining the double swab method to into this experiment, it could give me more of an accurate result than splitting the wet and dry apart. So.
All right, great, thanks. Excuse me. Anybody that has joined us recently, if you have any questions about Joseph's poster or missed his introduction, just let us know in the chat and we'll get those answered. We'll give everybody a few minutes to just read over the poster, and then maybe Joseph, you can go over your introduction again for anybody that's new to the room.
Hi, everyone. For those of you who have just joined the room, I'm going to start the presentation again. So my name is Joseph Rahm, and I'm a graduate research assistant at FIU, working under Dr. Dieta Mills on an enhanced collection and recovery method from smooth and rough surfaces for touch DNA. So as the sensitivities of DNA technologies continue to increase, forensic laboratories are expected to yield more probative DNA profiles with less and less collected biological material from crime scenes. The most commonly encountered biological material is known as touch DNA, and this is composed of epithelial cells left behind in oils and sweats on the hands of individuals, which is then transferred to surfaces and objects. Touch DNA, although a very powerful tool, given our sensitivities, has also presented many issues with their traditional DNA workflow. One of those being the inadequate collection by the standard method, cotton swabs, and the loss of DNA during extraction. So as I think about an approach to these problems, how to create a solution, we have to think about the factors that affect the collection of touch DNA across surfaces and how its relationship, the direct relationship affects the DNA profiles produced. And this, what I'm focusing on here is the roughness and porosity of the surface. So there has been intense focus on the downstream processes of the DNA workflow, such as increasing extraction efficiencies and lots of different kinds of polymerases and amplification kits. But there is still a gap that exists, and that is the front end process of collection. Now, there are practical reasons why we like cotton swab collection. It's fast and easy. The sampling method is cheap. But cotton does present two major issues, and one of those being cells become trapped in the cotton matrix and are not released effectively during extraction. And the other being that cotton rips and tears on rough and porous surfaces, especially when the swabs are pre-moistened. It is also worth mentioning at this point that swabbing surfaces for touch DNA with any matrix, not just cotton, is destructive toward other forms of evidence that may be present, like fingerprints. And so as I move forward with vacuum collection, I've kept that in mind so that one day I may be able to optimize this for other forms of evidence. So let's just jump right into how or what I did in the materials and methods section. So first, I want to introduce the vacuum that I'm talking about, the vacuum Venturi device, which is really this little black thing here that you see me outlining with the cursor. It is a fancy pump that you can put a compressed gas source through it, and it generates a suction because the choke of the pipe decreases the pressure. And I use that suction to collect DNA off of surfaces. So I connect the vacuum to a series of tubings and then connect a sampling syringe that is completely disposable, and I outfit alternative collection matrix that attaches to cells as opposed to absorbing them and trapping them inside. And this is the polycarbonate filter, which would fit on the end of the syringe here on the white suction cup. So to mock the touch DNA samples, I took buccal swabs from my cheek, and then I put the swabs into a solution. The cells came off, and then I calculated the concentration of that cell suspension and pipetted approximately 100 cells onto each surface that I collected from. So I then compared the vacuum collection with the cotton collection, and I did wet and dry treatments for both collection methods, replicates of 3 per each treatment. So then I got to a point in this workflow where I would have to extract my samples, extract my DNA. So because of the losses that are seen with touch DNA when they go through standard extraction methods, I decided to evaluate my collection methods using direct PCR. So using direct PCR and 6 core STR loci, I generated a mock DNA profile, and I plotted the results to the right here on this graph. So if you look at this graph, you see on the x-axis, the cotton and the polycarbonate filter being compared by each loci. So for each grouping of bars, there is 1 loci, D5, D13, D7, D16, the CSF loci, and penta-D loci. For each loci, you see our 4 collection treatments. Green is the cotton dry, yellow is the cotton wet, the teal bar is the polycarbonate filter and vacuum dry, and the blue bar is the polycarbonate filter and vacuum wet collection. So if you look at the y-axis, you see the height and relative fluorescence units, and you see that most of our loci for each treatment are clearing our threshold, which is denoted by the red bar of 150 relative fluorescence units. Some things to note on this graph would be that the wet and dry cotton collection do appear to have been outperformed by the polycarbonate filter vacuum collection, although the dry cotton collection did appear to produce higher peaks than the wet cotton collection, which was surprising to me. As you can see, for the polycarbonate filter vacuum collection, both treatments yield fully represented profiles with very high peaks. So as I move on with my research, I hope to optimize this method on rougher substrates, notably cinder block and different types of brick. And then hopefully as I navigate these 2 extreme collection substrates, I can move on to other porous complex materials like rope, synthetic or natural. And that is pretty much it. And with that, I'll take another round of questions.
All right, great. Thank you. We did have a couple questions come in as you were speaking. The first one is, do you have a cost for this equipment in relation to the Venturi vacuum and any consumables that may be required? And then they kind of had a tag-on question, is the tubing a one-time use only?
So those are great questions. The front tubing, that is a one-time use only, along with the syringe. I throw those away and make them new, sterilize the materials, autoclave them for every collection that I do. One thing about the cost, I'm honestly not sure. I developed this project from things that we already had around our lab. So we kind of just already have a lot of these things, and I haven't kept track of the cost in that way. In terms of consumables, with the filters themselves, the polycarbonate filters are about $70 per 100 filters. Those are a little bit more costly. But the tubing and everything and the syringes, they're all very cheap. We've ordered them in bulk, and I've just had them, a lot of them that I haven't ran out of. Was there another part to the question? Did I answer everything?
Yeah, you answered. Yeah, I think you answered both.
Oh,
Um,
in terms of also the solutions for collection, um, that is pretty costly. The DNA grade free water, but it's the cheapest cost when you consider taking the samples downstream and everything.
Great, thanks. We do have another one here, and they're asking, is it easy to extract from PCF, meaning does it break down?
It is easy to extract from. You don't really need to break it down. It won't break down completely. In a traditional extraction, you do see like the filter left over when you when you empty the cartridge or whatever method of extraction you're talking about. But when I do a robot extraction, for example, I will prep it just like I would a swab, and it's still there after the extraction. It doesn't break down completely like cotton swabs do, but we don't need it to because the cells are adhering to the surface. So when they go through extraction, they do release a lot better than cotton, but still, when I take polycarbonate filter and cotton through robot extraction, neither one performs well. So there's something about the threshold and the robot extraction that I'm doing that is not optimal for touch samples, which is why I did direct PCR, because even at this low threshold, I'm losing a lot in the extraction washes.
Thank you for those questions. Any other questions? I saw a couple people just come in. If you have any other questions about Joseph's poster, feel free to put them in the chat either to myself or to Joseph, and we'll get those answered for you. Joseph, we have one question here, and it is, how many replicates for each collection treatment was completed?
So I did 3 replicates per collection treatment, and I did a negative and positive control for each treatment.
And you kind of spoke about this earlier, but since we have so many new people on right now, can you talk about your results on rough surfaces again?
Sure, yeah. So my results on rough surfaces are not optimal to be shown. So I know there's a lot more in the optimization that I could have done, so I didn't show them here. But what they would have shown, my current results, is that as you move from smooth to rough surface, the difficulty, especially when the rough surface is porous, like brick, um, is increased. And so I've had to go through a lot to optimize on the brick by increasing the amount of solution that I'll use in a wet collection. But that also, there's a trade-off in that, because if I use too much solution to do a wet collection from brick, then I am risking the solution going back up into the tubing and into the vacuum and contaminating it. So I am working on optimizing that in a way that will not contaminate my device that I'm using.
Wonderful, thanks. Yeah, any more questions? Feel free, let us know. A question did just come in, Joseph, and it says, how could you repeat the collection portion that was done?
So for the collection, we're just going to start out with dry and then move on to wet. So first, to mock the touch samples, I took buccal swabs, and then I took those swabs and put them into a buffered solution, and they all came off the swab. And so I calculated the concentration of the cell suspension so that I could deposit approximately 100 cells onto each of the surfaces I was collecting from. Um, and then I allowed those cells to dry. And for the dry collection, I just went in with the cotton swab or with the vacuum and collected after they had that time to dry. Um, but for the wet collection, I allowed it to dry and then pre-moistened the cotton swab, um, and performed the wet collection like that. And then for the wet collection with the vacuum, I did not put the solution, or which was approximately 20 microliters of DNA, or no, I'm sorry, it was 20 microliters of DNA-free water. I put that directly onto the surface instead of the polycarbonate filter, because the polycarbonate filter is not absorbent at all. It doesn't take liquid, it just slides right off. So when you do wet collection with the polycarbonate filter, the moisture is applied to the surface, the collection substrate, not the collection matrix. And that's pretty much how I did each type of collection.
While you're explaining things, could you explain the vacuum as well?
Sure. Okay, so the vacuum Venturi device. The Venturi device is the little black pump that, hold on, let me show you the little black pump that I'm circling with the cursor. So it allows for you to shunt a compressed gas through the pump, and when you do that, the compressed gas goes straight through the pump as air, which goes straight out the other end. But as it's moving through the pump is being constricted in the choke of the pipe right in the middle, which decreases the pressure inside the pipe and generates a suction, which we then utilize with this blue tubing here to collect our touch DNA off of surfaces.
Joseph, one question that just came in is, do you know where you can purchase the Venturi vacuum?
Yes, the Venturi vacuum can be purchased from a website called Vacon. If you look at the little Venturi device in the picture here, you can see the words Vacon on the pipe. So you can get that from that website, and you can also get the the tubing and some suction cups and kind of everything on that website is geared toward using this to manufacture and clean computer parts, and that's kind of where this all comes from, the vacuum device. But using it in this context is a new thing.
One question that's being asked, Joseph, is how large is the Venturi vacuum device? Is it portable enough to bring to a crime scene?
So the actual device, the little black thing, is very, very small, but the entire apparatus, I'm assuming, is what they're asking about, which is the syringe and the tubing and the little pump hooked up to the CO2 tank. This is very portable, although not this tank in the picture. We have other size tanks in the lab, and the smallest tank is like a 5-pound tank that you could use it for like paintballing and stuff like that. But if you do decide to go with portability for a device like this, you might run out of air at the crime scene. So you kind of have to pick a size that you know would be okay for wherever it is that you're going, and kind of like for whatever crime scene you might encounter. You'd want to think about that before taking it out.
All right, very good. Another question that came in is, how does this compare to the Mvac?
That is an excellent question. So for the MVAC uses a lot of solution. The MVAC uses up to 5 milliliters of what they call filtrate solution to collect a single sample. And so this is more applicable to like collecting DNA evidence from the entire flooring of a car per se, where that might be necessary. But the Mvac in this scenario, it uses um, too much solution in the wet collection. And so there's a lot that you have to do to get that volume of solution down to a volume that has enough good DNA in it, the purity of it, to be able to use it. So the advantage of this device is that you don't have to use that much solution to collect in a wet collection, whereas with the MVAC, you have to use milliliters worth of solution to do one collection. I
Great
like that
question.
question.
Yeah. All right, one more question here, Joseph. Did you do a validation comparing the MVAC to this device?
No, and the reason for that is because we are not allowed to. We're funded by the NIJ, and they told us we could not do that.
Makes sense. Have you generated any data to assess the ability of the vacuum to collect cell-free DNA?
Oh, that's my favorite question of the session. So I have been thinking a lot about that, and this is my master's work. I will be doing that for my PhD, which I'll be starting later in the fall. But I realized when I was starting this work that there was no accounting for cell-free DNA in my mock touch DNA samples, that I could only rely on the DNA inside the nucleus of every cell. But I knew that there was a gap there, and that gap, to me, it can only be answered with a larger project. I don't think it could be answered in this project. But my approach to answering that question would be to find a way to, or some type of intercalating dye that would bind to cell-free DNA. And the problem in finding that kind of thing is that cell-free DNA is very fragmented traditionally. Um, so dyes typically need longer fragments of DNA to bind to. I have thought a lot about that though.
Another question that was just asked, and you may need to just do a little bit of clarification on this, is what they said is, of your 100 individuals, did you see any difference in DNA yield, shedder versus non-shedder?
For the 100 individuals thing has not been done yet, but we will be looking at that. The COVID situation really threw a wrench into arranging everything, and all of that was supposed to be done at the end of the spring, and so a wrench got thrown into that part of the project, but it will be completed as soon as possible, and we will be paying attention to that, because cell-free DNA also, from what I've read in the literatures and the mechanisms, the cellular mechanisms that lead to cell-free DNA being present in bodily fluids, suggest that the mechanisms for cell-free DNA are more complicated than just a bunch of chopped up DNA. So they typically can use cell-free DNA in bodily fluids in like cancer research and clinical laboratory science. And I'm currently trying to find a way outside of this project to bring the value of cell-free DNA that has been realized in other fields into this context. Does that answer your question?
If you have a follow-up to that, just let us know. One person was asking if they could get a copy of the poster.
I will have to check with my boss, but permitting that she says yes to that, I will send you a copy of it for sure. Just leave your information and I'll be glad to do that.
They're asking what studies have been done linking cell-free DNA quantity to shedder versus non-shedder status.
I have not read any studies that have linked the quantity of cell-free DNA to shedder status. Although I think there are a lot of environmental factors that contribute to shedder status. Like, for example, you may not be a shedder like in the middle of Michigan, but you may be a shedder down here because it is so humid. There's also the factors that shedders typically, there have been studies that cell-free DNA is shed in the bodily fluids of individuals who have specific cancers or specific autoimmune disorders or diseases, but there has been nothing, to my knowledge, connecting that to forensics, and that's kind of my goal is to connect all these interdisciplinary things, because I think that cell-free DNA is definitely connected to shatter status, but again, shatter status is not that simple. There's a bunch of environmental factors, including your own health, that contribute to that.
Joseph, are there any things in the future that you are looking forward to be able to try and do or get more answers on after going through this process?
Yes. So I'm looking forward to wrapping up this project with the 100 individuals on actual touch samples, but also to figuring out how cell-free DNA contributes to a touch DNA sample. That's something I'm definitely interested in, and um, I do plan on looking at that application in my PhD.
Great. So there you go. We got some things to look forward to. Joseph, one question. Can you talk a little bit more about why you chose CO2 as your compressed gas, and would there be other options that are available or better?
That's a great question. So the reason why I chose CO2 gas, I kind of just stumbled upon it, but there is, in trying other alternatives, definitely a reason why I prefer it over other things. And one of those other things that I tried was, if anyone is familiar with like lab settings, there's just, there's spouts for gas and spouts for environmental air, and so I tried to collect using that air spout, but it doesn't produce a high enough suction velocity to make it happen. And then I started reading into using alternative sources to generate that suction could present a contamination problem. So because CO2 gas is contained inside its tank, it's sterile, there's nothing that can, to my knowledge, affect the quality of the gas or anything that might mess up the DNA profile. We have to think about those things, especially when we put these things into practice in forensics. And so I just stayed with the CO2 because it is a sterile compressed gas source. It's relatively cheap, and I'm just comfortable with it, knowing that it is contamination free.
Great, thank you. So now's the chance to get your last minute questions in. If you don't have any, I would suggest that you head back to the main webinar and get ready for that. Thank you so much for joining us.
Thank you. Have a great day.